Over the last 20 years, our understanding of cancer has accelerated at just stupefying rates. Major advances in molecular biology, computer science, including AI, have advanced so quickly that no one lab can stay apace. Can't do that anymore. The only way to get the job done is through team science, and that's what the 4 scientists in the Find the Cause Consortium are all about. So now we'd like to give you the 50,000-foot view of what we do and the kind of progress we've made in the Science Consortium in the last year or so. So I'll start with my quarter of the puzzle. So my lab believes that the immune system is a key to defeating cancer. And the one thing that all types of cancers have in common is their ability to beat the immune system eventually. If we can fix that, we can prevent virtually any type of cancer. The immune system is the one biological system flexible enough to adapt to cancers as they arise and as they mutate. In fact, your immune system probably has already saved you from getting cancer by killing precancers you didn't even know you had. My lab is working with Gail, Charlotte, and Stefano to understand these immune responses and find ways to boost it. Now, as you can see on the first slide, the immune system is constantly protecting you against cancers that can be birthed anywhere in your body and that occur more and more frequently as you age. But most of the time, the immune system wins these battles, and you never know that you had cancer. But cancers are clever. As they emerge, some cancers produce compounds that completely shut down the immune system. Indeed, many of these cutting-edge immunotherapies that you've heard so much about lately target these immunosuppressive factors. They're really important. Through an unfortunate twist of fate, however, many man-made pollutants mimic these natural suppressive compounds and protect the emerging precancers from immune attack. In addition, many of the pollutants can simultaneously drive normal cells to become cancers and for relatively benign cells or cancers to become crazy, aggressive metastatic killers. So it's kind of a double whammy. Pollutants can initiate cancer while suppressing the one biologic system that can beat virtually all cancers. There's a simple formula for the risk of this happening to you. The greater your exposure to these pollutants, the greater the risk. Now, what my corner of the Find the Cause Consortium has done in the last couple of years is to work out ways to boost the immune system for cancer immunoprevention. We've identified receptors that both the cancer-derived compounds and environmental chemicals use to suppress immunity. From there, we synthesize non-toxic drugs that blind the receptor to these immunosuppressive chemicals, therefore boosting immunity and in some cancer models, completely preventing cancer. I would like to emphasize that what we think are very important advances were possible only because funds from Find the Cause have allowed us to take chances, to do some very high-risk, high-return experiments, the kinds of experiments that are unlikely to be supported by, for example, the NIH. Now, even if we have discovered strategies for blocking the acute effects of pollutants and pre-cancer-generated immunosuppressants, we still have to know exactly when to initiate the interception. When do we actually begin to interrupt the process? That's where Gail and Charlotte come in with technologies to estimate carcinogen exposure and to predict when something bad is about to happen. Gail, why don't you explain your part of the puzzle here?
Thank you, David. My lab is developing a rapid test to determine whether a person has been exposed to unsafe levels of an environmental carcinogen, which can cause cancer. And we're looking specifically at a family of small signaling molecules called microRNAs. Now, in your body, there are thousands of microRNAs, and their role is to control normal function, normal processes in your body. And these are messengers. They're made in one tissue, and they can go to another tissue via the bloodstream, and they profoundly change and regulate this host tissue. Unfortunately, when we're exposed to carcinogens, they cause abnormal microRNA production. And this abnormal microRNA production can lead to either the induction of a gene that will promote the cancer or the inhibition of a gene that would have reduced the cancer. And that can happen in your breast tissue. And that's how this carcinogen is working, how it works to convert your normal tissue to a cancer. So we're using 3 approaches that identify the changes, and I'm going to focus on just one, which is a mouse model that will allow us to see what changes are happening as a result of carcinogen exposure. And this experiment is being done in collaboration with David. So the concept is simple. We are attempting to mimic in the mouse the process that happens when a woman is exposed to an environmental carcinogen. The complex of the carcinogen and its receptor will activate abnormal microRNA expression, and we're able to monitor that in the laboratory by looking at what is in the tissue that's been affected and what's in the bloodstream. And these changes that we're detecting happen well before the tumor appears. So we are monitoring changes in the microRNA, and what we have seen is that there's a group of microRNAs that are being induced as a result of the carcinogen treatment that appear well before the tumor appears. And that gives us a window of opportunity, if we can measure these microRNAs or when we measure these microRNAs, to alert a person that she or he has had an unwelcome exposure to an active carcinogen. Now, we're in the process currently of measuring which ones we want to use in our panel for microbiomarker exposure. And in this effort, we're being greatly helped by Stefano, who's helping us sort out using computational techniques to identify the specific panel we're going to use. So this assay can inform a person that they have received a dangerous exposure in a time frame that allows them to either prevent the exposure, to identify what could be the exposure and prevent it, or to intercept it early in the process, well before cancer arises. Thus, the information can affect negatively the impact of progression, allowing time to reverse and stop the cancer. This is the objective of our work, and we are beyond grateful to find the cause and the donors and supporters such as you that are generously providing the funding for these studies. So let me introduce Charlotte, who will show you an amazing video of cells, that mammary glands that are forming before your very eyes in vitro, and the power of this amazing new model to understand how carcinogen exposure and genetics intersect to cause breast cancer.
Thank you, Gail. I wanted to continue on about what she's been referring to. Through the years, my lab has been developing technologies that we can use to understand and study the biology of human breast development and use this to understand the process of cancer formation. One of the things that we've really been focusing on over the years has been identifying which cells in the breast give rise to cancer. But now we've pivoted a little bit away from using animal models and these approaches to studying this, and moving now towards tissue engineering, where we can actually create miniature human breast tissues in the lab. By using donor tissue from women that undergo elective surgeries, we can isolate single human breast cells and put them into a specific three-dimensional gel that enables the cells to divide and actually form a functional human breast. In this video, you can actually see this taking place in real time. A single human breast epithelial cell begins to divide and self-assemble into a small ball of cells. This ball then extends out and forms these long branch-like structures that invade into the gel that are driven by the tips. As the structure keeps growing, the branches get larger and get wider, and there's a lot of cellular trafficking taking place within this structure. At one point, you'll see buds starting to appear at the end of these ducts. These buds get larger, they grow out until the whole tissue begins to mature and form what we refer to in the breast as a terminal ductal lobular unit. You can think of this as a cluster of grapes, where each grape represents a small little ball where the milk is produced, and they're all connected to the stems, which are the ducts. What's really fabulous about this is that we can treat these cultures with hormones and they can produce milk. So we've really been able to successfully recreate mini human breast tissues in the lab. So we're using this technology and this engineered human breast tissue to study the effects of environmental chemicals on not only normal tissue growth in real time to see whether or not there are actually changes in the tissue as it grows, but also to see whether or not there's abnormal growth in response to these chemicals, being potentially precursors to cancer or even potentially overt cancers. We're studying a class of chemicals called endocrine disruptors, including the bisphenols, which you probably heard of, the BPAs, but also the larger class of forever chemicals. And we're looking to see whether or not exposure to these chemicals affects the tissue formation and also allows it to adopt a more precancer type of tissue. We're also using chemicals that induce DNA damage, like carcinogens found in cigarette smoke, and other DNA damaging agents like irradiation, to see whether or not they can actually cooperate with these chemicals to accelerate the formation of cancer. So an advantage of this approach and this platform and this technology is now we can see what's happening in the tissue in real time in response to these chemicals, something we've been unable to do previously and certainly been unable to do in the context of human tissues. We can now also isolate these cells from these tissues and actually examine at a very molecular and genetic level what effects took place in those cells as a response to those chemicals. By collecting the DNA from these cells and sequencing them, we can look for mutations. We can also look for epigenetic changes that decorate DNA in response to chemicals and see whether or not certain chemicals have different effects on the breast tissue. You can imagine this type of approach, while is very exciting, generates incredibly huge data sets that need to be interpreted not only to identify the mutations and the specific genes that are being affected, but also in a larger context to understand more molecularly and globally what these effects are having on the cells. And so this is where working with Stefano's lab becomes a real valuable asset to the consortium because of his expertise and the various assays and techniques he has to interpret large-scale data. We'll now be able to identify and ideally find what these exposure fingerprints to these various chemicals look like and overlay that with the actual changes in the biology and the tissue structure we see in the lab.
Thank you, Charlotte. Fascinating work. And indeed, we live in a data-rich age, as we all know, and the ability to extract knowledge from it is becoming essential. And so let me start by giving you a bit of a background here. I studied cancer for decades, but perhaps the work I'm most proud of is what we named the Carcinogenome Project, which was in part made possible by because funding. In that project, we combined computational and experimental approaches to develop methods for the identification of new environmental carcinogens. As you've heard, carcinogens are basically cancer-causing chemicals. So the approach that we developed achieved an accuracy of about 80%, which is, it means that it identified a carcinogen correctly about 8 out of 10 times. That's pretty good, but there's, of course, hope to do better. And in fact, there is a recent study, which in part builds upon our work, but independent of our work, where they applied AI, artificial intelligence methods, to new and larger data sets and achieved an accuracy of above 90%. That is 9 out of 10 times they were correct. And this is amazing progress, and it points to the importance of sowing the seeds that will encourage this type of research, which is clearly prevention research. And for the reasons David alluded to before, we had a much harder time securing federal funding for it. And that is where actually Find the Cause became indispensable, as it helped fill that funding gap. Now, working again together with Dave, Charlotte, and Gail, we're shifting our focus from the identification of harmful chemicals to the detection of exposure to those chemicals in humans. So as you've heard, Dave, Charlotte, and Gail's experiments are helping to identify so-called biomarkers. Basically, these are sentinel molecules, genes, microRNA, as Gail alluded to, epigenetic marks that are activated in response to exposure. And we want to use these biomarkers to develop computational predictors of exposures in tissue and in blood. And why is this important? Because if we can determine early that a person has been exposed to potentially harmful chemicals, we can, one, try to remove the source of that exposure. Two, we can monitor more carefully the exposed individual for early signs of malignant transformation. And three, we can recruit their own immune system to fight the harmful effects of the exposure. That's what David told you about. So hopefully you can see how the four pieces of the puzzle, that is the Find the Cause Consortium, fit together to better understand how cancer happens, to be able to screen for biological changes that may lead to cancer, and to find ways to intercept cancer before it happens.
Yeah, I agree with all of that 100%. One of the things that comes to mind that really excites me now is what you can see on the video that's just started. It's a visualization of the length of our reach out into the scientific community. And what you're seeing is the institutions and individuals that have cited our work in any given year throughout the whole world. And you can see that we're making an impact pretty much everywhere. So now you all can see why we're more than a little obsessed with finding the cause and preventing cancer. We hope that you, our viewers, are motivated to do what you can do to help find the cause, not just us, to help you and your kids.